In this final lecture, I turn from the senses themselves, the senses of man and animals, to the brain, to the nervous system, to that magical computer that we carry in our heads, the computer that interprets our senses. This lecture is about the extraction of meaning from the senses by the brain. The brain may have started in evolution as a very simple form of connection between sense organs and muscles, between senses and effectors, but it's become so much more than that, a magical instrument that goes beyond the senses. Sir Charles Sherrington, when he was Professor of Physiology earlier this century, wrote about the human brain at a stage when knowledge about the brain really wasn't very advanced. But he, with great insight, imagined the brain like an electronic instrument with flashing lights and sparks. And he imagined the brain of a waking man. And he wrote, "The brain is waking, and with it the mind is returning. It is as if the Milky Way entered upon some cosmic dance. Swiftly the head mass becomes an enchanted loom where millions of flashing shuttles weave a dissolving pattern, always a meaningful pattern." Well, we'll see in this lecture the way that the brain does extract meaning from the senses. It is an instrument of intelligence, and our senses would be nothing without the ability of our nervous system to interpret them. Well, how did people first start thinking about the organization of the brain and the organization of sensory systems within the brain? This model shows an early attempt from the start of the last century to describe function in the brain. It's a so-called phrenological head. The theory of phrenology was introduced by Gall and Spurzheim at the beginning of the last century. Although they were excellent anatomists, their scientific methods weren't very good because they decided that they could study the organization of the brain just by feeling bumps on people's heads. And they went around feeling the heads of their friends, asking them about their character and their interests, and suggesting that each bump represented a particular function of the part of the brain below the bump, the part of the great cerebral hemispheres that lie directly underneath the skull. And you can see from this model that they described peculiar functions like sublimity here, and ideality here, and cautiousness here, ascribing them to different parts of the brain. They called them organs of the brain. Well, that concept of how the brain worked was really a very spurious one, and it fell out of favor with scientists very quickly, although it persisted in popular science until the first half of this century. This model was made only about 30 or 40 years ago. But the important step that Gall and Spurzheim had made was introducing the idea that functions were indeed localized in the cerebral hemispheres of the brain. Well, we certainly know that that is now true, and we know it first from the work of neurologists, of clinicians who looked, starting in the middle of the last century, at the effects of damage to the human brain. They found that after local damage caused by an injury or sometimes by a stroke, by bleeding into the brain, people could have certain sensory functions affected. And different damage would produce different effects on each of the senses and different aspects of perception. And from their work and many other sorts of scientific techniques, we now know a great deal more about the true localization of function in the brain. This is a model of the human brain. Here's the front of the brain, and this is the back of the brain. We now know that the entire back half of the cerebral hemispheres, all of that, seems to be devoted virtually exclusively to the analysis of vision. Vision's obviously very important for us. And here, further forward in the brain, is a strip of cortex that analyzes information from the body's surface. And lower down on the side here, an area that deals with sound. About 2/3 of the entire surface of the cerebral cortex of man may be devoted to the senses. Let's look now at a very modern technique, again that's used by clinicians working in hospitals to study the localization of function in the brain. This first slide shows the research laboratory of Dr. Marcus Raichle in St. Louis in the United States, and he's using a very modern X-ray technique that's also used at the Hammersmith Hospital here in London, called positron emission tomography. The method was developed in this country. It's based on the extraction of information about X-rays within the brain from a very sophisticated X-ray scanner placed around the head, in this case the head of a normal human volunteer. A computer interprets the signals from the X-ray machine and builds up a picture of a slice through the human brain. The next picture shows the principle of this process. The X-ray machine extracts a view of one thin slice of the brain. Now, if this technique is combined with the introduction of a radioactive material into the blood of the subject, then wherever blood is circulating through the brain, radioactivity will be localized by this instrument. Now, where does blood circulate? It circulates where there are active cells, including active nerve cells. So any highly active part of the brain will become labeled, radioactively labeled, for a short period of time during this method. Now let's see what happens. The next picture shows an X-ray processed by the computer from this PET scan, positron emission tomography. Here's the human brain, that's the front, this is the left-hand side, that's the back. It's colored by the computer according to the level of radioactivity. And at the moment, the person is lying in complete silence, and there's very little activity going on in his brain. But then for the next picture, we see an enlarged view of that place. Here it is, the side of the brain which we know deals with sound. Still in silence, we see that there's very little activity in the sound area of the cortex. Now, the patient lying there has played to him a tone of 400 cycles a second for a few seconds. And the next picture shows what happens. Here suddenly is a localized hotspot of activity, many, many nerve cells all firing furiously, labeled by that red spot of high radioactivity. Then the pattern's changed, and the subject is played a tone of 5,000 cycles, just a slightly different tone. And this is the next picture. Instead of the activity being at the front, the activity has now moved just a short distance away to a slightly different place within the sound area of the cortex. So we see then that within each of these regions of the brain, where different senses are represented, there is a kind of map or representation of that sensory system. If we were to look in the human touch area of the brain, we'd find a very peculiar picture or map of the surface of the body. Now, do you remember in an earlier lecture I demonstrated the way in which the skin of the fingers is very much more sensitive to detecting the separation of objects than the skin of the arm or of the back? That's because there are far more nerve fibres going to the skin of the finger than to other areas of skin. Well, this means then that the part of the brain receiving information simply has more information to deal with from the fingertips, and actually from the lips, than anywhere else on the body. So the form of the map of the body is a very distorted one. In fact, it looks like this. This peculiar statue is a model of the map in the human touch cortex. You can see that the thumb and the fingers, the lips are very big, and the body is squeezed up and tiny. It represents the scale of the map. There's much more cortex representing the parts of the body that have many nerve fibers than those parts that have very few. So these maps are very odd representations of the body. They reflect the function of each part of the body. Well, the importance of different parts of the body varies from animal to animal. Our fingers are terribly important because we pick things up and we touch with them. Many other animals don't. Can I have the next visitor, please, Bill? Now, I think perhaps you ought to hold. Okay. This is a friend from the London Zoo, and I should take this opportunity to thank the London Zoo for supplying so many animals during these lectures. This, of course, is a pig, a baby pig, and you can see that he has a very active snout. This part of his face is extremely richly filled with nerve fibres, and he uses it to hunt around on the ground in much the same way that we use our fingers. Thank you very much, Bill. Now, when Lord Adrian in Cambridge, who was professor of physiology, when Lord Adrian in Cambridge started to study the brain of the pig, the touch area of the brain, he found a quite remarkable sort of map. The next picture shows Adrian's map of the pig's brain. Down at the bottom part here, we can see the side of the pig's brain with the visual area and the hearing area and this very large touch area. But when Adrian recorded electrical activity from that region, he found that all of it was receiving signals just from the snout. The rest of the body had virtually no representation at all in the pig's brain. So the most important, richly innervated part of the body occupies the biggest space. Now can I have the next animal, please? Now this is a very different little creature. A friend, the mouse, we've seen lots of times. Now look very carefully at the face of the mouse. You can see the whiskers on his face, and you see the way that they're twitching constantly all the time. He's whisking away, searching the ground in front of him. The whiskers are attached to a pad on the side of his face, a pad which is simply full of nerve fibres picking up signals from those whiskers. Well, we might expect then, if we were to look in the brain of a mouse, to find that the whiskers, like the pig's snout, occupied a very large part of the body map of the mouse. Well, that's certainly the case. If we look at this next picture, this is the brain of a mouse. The brain of a mouse, and you can see on each side of the brain, we're looking from below the brain, two particular nerves, these white things here. Now this thin little thing here is the meagre little optic nerve coming from the eye. That surely can't be terribly important to a mouse. Whereas this huge, great tree trunk here is the nerve that innervates those whiskers. Probably much more important than the eyes. Now, when Hendrik van der Loos and Thomas Woolsey in the United States looked at the structure of the body area of the mouse in the brain, they found, not surprisingly, that the whisker representation covered a large part of that map. And as they cut their microscopic sections through the structure of the brain, they suddenly saw a most extraordinary picture. The next slide shows it. This is a single section, a very thin microscopic section, cut through the brain of the part of the brain that represents the body of a mouse. Each of these little tiny blue dots is one nerve cell sitting in the brain, waiting for information to come in from the body. What van der Loos and Woolsey saw was these rings, these dense rings of nerve cells, which I think you can see quite clearly, forming a sort of cobblestone pattern, a mosaic across the brain. Each of these things is just a small fraction of a millimetre across. Well, when Woolsey and van der Loos looked at the picture, they suddenly realized that this pattern was identical to the pattern of whiskers across the side of the face of the mouse. There's literally a picture of the whiskers inside the mouse's brain. And each of these rings, or barrels as they're called, receives all the nerve fibres coming from one of the whiskers. So each animal then has a map of its sensory world, and a map which is distorted according to the importance of each bit of the sensory world. What on earth could be the function and the meaning of those maps in the brain? Well, you know, when psychologists first discovered what the neurologists had found out, some of them even suggested that the maps might be perception. That just the existence of a pattern of activity on the surface of the brain might be the way that we see or hear or feel things. Well, that can't be right, can it? And it was recognized as being wrong even in the 17th century by René Descartes, the philosopher. You'll remember that Descartes had discovered, along with others, the retinal image in the 17th century, and his picture of the organization of the brain introduced the idea that there might be pictures in the brain. Here's Descartes' very fanciful view of how the brain was organized. You can see the two eyes looking out at an arrow in space. forming their nice little image of the arrow. Then the nerve fibres from the eye went in to the sides of the ventricle, those magic fluid-filled cavities of the brain. Inside the ventricle was a small little organ called the pineal gland, whose function we still don't know. Descartes suggested that this pineal gland was the seat of the soul, or the mind. Now you can see what happens. As the activity comes in from the eye, it sets up a pattern of vibration in the walls of the ventricle, which somehow projects onto the pineal gland to produce a little picture of that arrow in the outside world. Well, that's all very nice and an amazing piece of speculation on Descartes' part. But as Descartes himself said, although this little picture still retains some resemblance to the objects from which it originates as it passes to the back of our head, we should not fall into the error of thinking that it is by means of this resemblance that we see the objects, for there is not another set of eyes inside the head with which we could see these things. You see the point? It's not just enough to produce a picture. There is already a picture inside our eye, on the retina, but that doesn't create perception. The picture has to be seen. Well, the question, of course, is who does the seeing? Well, the seeing, if our present views are right, the seeing is done by the nerve cells of the brain themselves, the very cells which make up the map. Those cells are interpreting, are processing the signals that they receive. They are the elements of perception. Neurophysiologists studying the brain, recording impulses from nerve cells in the brain, have told us a great deal about the organization of these sensory areas and how the nerve cells in them are able to process information almost in an intelligent way. Perhaps the most exciting discoveries in this field came from two men, David Hubel and Torsten Wiesel, working at Harvard Medical School starting about 30 years ago. And in fact, they received the Nobel Prize last year for their work. They recorded activity from the visual parts of the brain in cats and in monkeys, and subsequently similar work has been done in many other animals. And what they found was that each individual nerve cell sitting there in the brain, receiving messages from the eyes, responded only to a certain sort of pattern placed out in space at a particular point in front of the eyes. Every different part of the brain was looking at a different part of the world, and every different cell was looking at a different thing in the world. Now, I'm going to play you a piece of film in a moment which shows an experiment of this sort being done in my own laboratory in Oxford. I'm recording electrical activity, and you'll hear the nerve impulses from a single nerve cell in the brain of a deeply anesthetized cat whose eyes are watching a screen, and you'll see that screen too. And I'm projecting patterns of lines on the screen. Look very carefully and listen and see just what kind of pattern this nerve cell responds to. I think with the electrodes in, sit down and start searching for responsive nerve cells, moving a pattern in front of the eyes, the animal's eyes, to see if I can activate any cells advancing the electrode as I go.
The bar of light is moved in front of the cat's eyes at various angles to see how the cell responds.
Getting a little bit weaker now. Weaker still. And now, virtually no response at all. The cell is just producing an impulse of its own every now and then, just firing off spontaneously. So the cell really responds only over quite a narrow range of angles of a line presented in the correct part of the visual field, and in this case, moving in the correct direction. You see the response to movement downwards is very much better than the response to movement upwards. So you can see this cell responds best for a line at a particular angle. Different cells prefer different angles all around the clock. I suppose you might look on this as a very first stage in perceptual analysis. Presumably these neurons in the cat's cortex are telling it something about the shapes of objects out in the visual field. Well, we almost certainly have similar sorts of nerve cells in our brain, in our visual areas, each cell responding to a different angle of an edge or line in our visual field. Presumably the way that I can see you and recognize this room is by breaking down all the component lines and edges amongst the nerve cells in my brain. So, nerve cells can behave intelligently. Certainly the brain as a whole is performing a fantastic process of analysis on sensory information. And that analysis goes beyond the information alone that the brain receives. We seem to have expectations about the world built into our brains, and we need only a little information from the sense organs to produce the right perceptions. Well, are they always right? Can I have a look at the next slide, please? This is the surface of the human eye, the retina of the eye. You've seen this picture before, photographed through the pupil. There's the fovea, the very sensitive area, and here's the blind spot, as it's called, the optic disc, where all the nerve fibres leave the eye and the blood vessels come in. Now, it's called the blind spot because in that region there are no photoreceptors. It is literally blind. We can't see with it. Well, if you'd all like to cover up one eye with your hand and then stare out in front of you, are you aware that there's a big blind hole somewhere out here at the side? It's about as big as your fist at arm's length, but you never see it at all, except with very clever tricks. There's an area in which you are literally blind, that you can't see anything, but you're not aware of it. Your brain somehow fills in that missing gap in the information because it has the expectation that there really are not holes in the world. Well, let's see how those expectations might sometimes go wrong. Could I have the next slide, please? Well, this is obviously a nice picture of a lady smiling. Quite clear what she's doing. Well, is it? Could we have the picture turned up the other way, please? Well, you can see she isn't really smiling properly, is she? Now, if we could go back to the previous picture, I think you'll see what's been done. The smile has been turned upside down. But at first glance, it looks quite reasonable. It's almost as if we pick out, we have a detector for, if you like, that smile pattern. We interpret it as a smile, and we don't bother about the relationship between the smile and the rest of the face. Maybe we have smile detectors in our brains, and the relationship between them, between the smile and the face, is based on the expectation that normally smiles lie the right way round on faces. Well, let me show you another example of the effect of expectation in a different sense, and I'd like to have two volunteers, if I could, please. Would you two like to come down? Would you like to come as well? Come on down, come and help. What I want you to do, what are your names? Isabella.
Judy.
And Judy, right? Isabella and Judy are going to help us to measure the weight of these two big snowmen here. Now, there's this big chap and that little one. Would you like to just pick it up and sort of estimate its weight? And you take that one, feel it, pick it up, feel how heavy it is, okay? Now change over and feel the other one. And I want you to tell me when you've felt them which one is heavier. Now, what do you think about that? What do you think? The small one's heavier, yes. What do you think? Do you agree? Definitely? No doubt about it at all? Well, let's see if we can weigh them and see them, shall we? Bill, would you like to take that one? We'll put them on a pair of scales and just see which is heavier. All right? Okay. Well, they seem to be exactly the same weight, don't they? Well, don't be worried, because everyone makes the same mistake. This is a well-known illusion called the size-weight illusion. We expect big things to be heavy, and we somehow scale our judgments of weight according to the size of objects. These two things actually weigh the same weight, but because we expect that the large one will be heavier, for some reason we scale down its apparent weight, and it feels lighter. Thanks very much. Thank you. Well, then we have expectations about the world which influence our perception of the world. Other animals have expectations too, although they tend to be, in many cases, rather simpler than our complicated expectations of the world. Let's look at the next bit of film, which shows the expectations of a toad. This film was made in Dr. Evert's laboratory in Germany, and you see a toad gobbling up a mealworm. Now, what sort of expectations does a toad have about mealworms? Well, here's the toad making exactly the same reaction to a simple, moving, horizontal black line. He thinks it's a piece of food. He's snapping at it. But now what happens if the line is made vertical? Exactly the same bar, turned vertical and moved in the same direction, produces no response from the toad at all, but the horizontal one does again. So the toad seems to have very simple expectations built into its brain, presumably built into the properties of its nerve cells. Its expectation is that horizontal, long things moving along are food. And the same thing, vertical, is not food. A rather simple piece of computing, but quite enough to keep toads alive. Well, our expectations about the world are obviously more complicated than that. Part of our world is made by us. We don't live in an entirely natural world. We live to a large extent in a self-constructed world. And the fact that we live in square houses and in buildings has influenced our perception and our expectation of the world. Now, I happen to know that elsewhere in the Royal Institution, Bill Coates is doing some quite amazing research, and we've got a chance to look in on his experiments, to be one of the first observers to see Bill Coates' anti-gravity experiments. I wonder if we could possibly go over to the other cameras. If you watch the monitors, you'd see the room in which Bill's working. Now, ready to do have you got the anti-gravity machine on out there, Bill? You see he's put a ramp way there, and he's going to put the ball on, and goodness, it's rolling uphill. Well, that's obviously a really rather magical room. I think I'd like to go and investigate that room, and to go there I'd like some help from two people we've seen before, Shireen and Zarina, who, as you know, are twins. Would you like to stand up? Now, we're going to go out and find the magic room, and you can watch us on the monitors. So come along outside. Okay, come along, Zarina, come on this side. There we go. Well, off we go, girls. Now, here's the magic room. Now, Zarina, do you think you'd like to go into it? Just go into the left-hand corner. Oh, that's very good. Now, Shireen, let's see if we can make you grow. You go into the right-hand corner. Well, my goodness, that's a magical change, isn't it? Perhaps you better change positions. Can you change over? Well, this is an odd room. It can make people grow and it can make people shrink. I think you better come out in case you stay that size. Out you come. Well, now we can bring the camera out and see what the room is really like, because it's out here. And you can see it's really a very odd room. It's not a square room at all. It's got very strange angles. And the reason why Shireen and Zarina looked such a peculiar size was because we had expectations about the shape of the room. Therefore, their size seemed to change as they walked backwards and forwards. Would you like to go back in, Shireen, and show us? Just walk into the corner. And then walk up to the side. You can see as she walks across, she occupies more and more space within the room, and therefore we think that she gets bigger if we view it from just the correct position. Thank you very much. Okay, off we go. Here we go. Well, you saw there in that demonstration the way in which a strange object could be made to look correct by viewing it from the wrong position. Artists, when they paint pictures, all the time face this problem of presenting on a two-dimensional surface a proper representation of the real world, if they're painting representational pictures. But this means that an artist's painting can only be viewed correctly and properly from the place that he painted it. The artist paints from one particular direction, what he's looking at, and therefore the picture should be viewed from that direction alone. And if you look at a painting from a strange position, it looks quite different. Well, let's look at a painting by Hans Holbein and see how that might work. This is obviously a very nice picture painted in the 1530s. It's called The Ambassadors, and it's in the National Gallery just a mile or so from here. It's of the French ambassador to the English court and a friend of his, and they're surrounded by nice objects, as you can see, except for this strange thing here lying on the floor. Now, what on earth could that be? It looks like perhaps like a French loaf or something. In fact, that's a joke of Holbein's. It's another picture, a second painting, painted from a different direction. Instead of standing in front of the painting, Holbein moved around to the extreme side to paint that picture, and we can see what he saw if we look at the other camera's view of it. Now, if you look at the monitors, you can see what this object is. I'll move the pointer backwards and forwards along it. Painting of a skull painted at this very strange angle because it was painted from the position where that camera is now standing. Well, that's an artistic joke from Hans Holbein showing how important it is that we judge what an object is from the position at which we look at it. Our senses, then, are trying to extract meaning out of the world, and the brain tries to interpret reality on the basis of sometimes rather poor information from its sense organs. What we need to know about the world is what is really there in the world, and sometimes our senses don't provide us with accurate information. The brain has to provide corrections. Let me show you an example of how the brain is able to correct for changes in brightness. I'd like someone to help me with this demonstration, please. Would you like to come down from the front row? Good. Now, could you just stand up, stand towards the camera, and hold that piece of card just about there? Yes, I think that's very nice. Now, quite clearly, this is a piece of white card and black card. There are no Hans Holbein tricks here. Absolutely straight, and I've got an instrument here which is going to measure the intensity of the light, of the brightness of those two surfaces. First of all, the white surface. It's about, let's change the scale, it's about 54 units of intensity, okay? And the dark one, of course, much less, about 39 units. 3.9 units, much, much less than the white surface. Now I want to turn the room lights down until the white surface is also 3.9 units, where of course it should look the same as that one does now. So could you try and remember the black one? Can we turn the lights down, please? 15, 12, 10, carry on. Darker, please. Okay, that's correct. The white surface is now the same intensity that the black surface was. But I think you'll agree they still look like black and white. Could we turn the lights up again, please, to convert the white one back to its original? And I think you'll agree that they don't appear to change. What we're judging is the relative intensity of the two, and we simply disregard changes in overall brightness. Thank you very much. Thank you. Well, the psychologists call the ability of our brains to extract reality out of strange information from the sense organs, they call those mechanisms constancies. And the best example of a constancy method in our brain is what they call size constancy. Obviously, as an object gets further away from us, the image in our retina of that object gets smaller and smaller, but we don't see it getting smaller. Somehow the brain applies a correction. It says, well, I know the size of the retinal image, I know how far away it is, therefore its true size is X. I'd like you all to do an experiment for yourselves. Could you hold one hand up with your arm bent like that, and then the other hand off to the side, twice as far away? Now, I think you'll agree that if you look from one hand to the other, they look about the same size. I mean, they're two hands, aren't they? But if you think about it, the one which is closer to you has an image on your retina which is at least twice as big as the other one. It certainly doesn't appear to be twice as large. Somehow your brain is correcting for the difference in viewing distance. Well, that's fair enough if you're looking at objects which really are at different distances, but it can lead to mistakes if your brain is fooled into thinking there is distance which isn't really there. Let me show you a demonstration of that. Out here, I've got two little Father Christmases, and as you can see, they're both absolutely identical in size. They're just exactly the same diagram. But if I take them off, I can get them off and place them in a different part of the same picture. And now if you look at them, I think you'll agree they're quite different in size. The size is changed, although this is a flat surface, the size is changed by the apparent distance of the two. This perspective diagram makes you think that this one is further away. Therefore, your brain, applying its proper program, assumes that that must be a bigger Father Christmas because the retinal image size is the same, but that one's further away. So in that case, your brain has been tricked into assuming differences of size by believing that there are differences in distance. Well, in making this magical estimation of reality, the brain has to deal with very difficult problems. Not the least difficult is this problem of delay. You'll remember that the sense organs send messages to the brain along nerve fibres, and those nerve fibres conduct rather slowly. So there's a very considerable delay of information between the signal reaching the eyes or the skin and reaching the brain. So we live in a world which is actually behind the real world. Our sensory world is always slightly out of date. Let's ask someone to come and help to see exactly how out of date it is. Is Julia up there? Yes, Julia, can you come down? This is my niece, Julia. She'll help us with this one. Now, could you come and sit in the chair, Julia? Could I have the buttons, please, John? So, Julia, you've got a button there that you can press. I've got one here. When I press this button, that's going to light up. A little flash is going to go on. And at exactly the same time, the oscilloscope sweep over there is going to start to cross the screen. Don't press yours for a moment. Just watch what happens. The light goes on, the sweep goes across, and you can see that it's deleted the little pulse that was there before. When you press your button, a pulse will appear on the screen at a certain time after the beginning of the sweep, depending on how quickly you react. So I want you to put your thumb on your button, ready to go. When you see the light go on, press your button just as quickly as you can. Are you ready? Off we go then. How long is that delay, John?
About 220 milliseconds.
220 milliseconds, just over a fifth of a second. Shall we try once or twice more? Are you ready again, Julia? Off we go. Ready? Very good. What was the delay? Oh, that took too long. And right, off we go. Let's try again.
That's a bit longer. It's about 250.
About 250 milliseconds. Once more. Ready? Good.
Oops, it's gone again.
It's gone again. Try once more.
Okay. Again, about 220 milliseconds.
About 220 milliseconds. Thank you very much, Julia. Well, you see, it was taking up to a fifth of a second for information to pass from Julia's eye to her thumb when she pressed that button. And what's more, it was a variable time, sometimes longer, sometimes shorter. Well, her brain has to deal with this constant and very considerable delay in information. Imagine you were receiving a service from Bjorn Borg at Wimbledon. The ball would be travelling perhaps at 70 miles an hour. At the moment that the ball hit your racket, you would be seeing it crossing the net. So you have to design your movements on the basis of knowing where the ball was a very long time before. We have this problem always of dealing with and predicting the world on the basis of out-of-date signals. Now, can I have another volunteer to help me with a demonstration of this problem? Would you like to come down there? Yes. Now, what's your name? It's Lorenzo. Lorenzo, would you like to sit down? Very simple task for you, Lorenzo. This is a ball. I'd like you to point with your finger at it, if you could. Just point your finger towards it. That's it. And every time I move the ball, I'd like you to move your finger as quickly as you can to follow it. Are you ready? You see the delay? He doesn't know when I'm going to move the ball, and it takes him a fifth of a second to move. But now look what happens. Just keep going. Now he's perfect. He's not lagging behind the ball. He's pointing right at it all the time. Now, what could be the reason for that? Okay, Lorenzo, that's lovely. Thank you. Well, the reason must be that when the ball was swinging freely, it was following a regular, predictable path. Lorenzo's brain, within a fraction of a second, calculated that predictable motion, and he was therefore able to subtract out the delay in his own brain and move his finger correctly with the ball. Only when the motion was unpredictable could he not, of course, have any way of knowing what was going to happen next. So our brain is able to deal with delay by making predictions. Well, there are problems, of course, when the delay varies, and there are also some nice, funny illusions that happen with variable delay. I'm going to demonstrate one of them to you, which Horace Barbridge. Now, the fact is that our eyes, the photoreceptors in our eye, have different delays depending on intensity. The rods, working in dim illumination, send signals much less quickly than the cones do. And even the cones alone send signals less quickly when there's not much light around. So if we look at a picture like this, could we have the lights down, please? If we look at this sort of picture projected on the screen, which consists of a bright part, parts that come from the dark parts, will be delayed more in entering our brain than signals from the light. Move a target, we will see some bits of movement before others. Now, I'd like you to look at this nice, solid, wooden, and see what happens to it as I move it. I think you'll agree it seems to be moving rather well. It's a nice, solid rod being bent by visual magic. Now let's try this one, just because of the Christmas theme. This is a Christmas angel. Let's see if we can make the Christmas angel flap its wings. I think you can see the edges of the wings going up and down rather nicely. Okay, back once more to the rod. If we could we turn the lights down once more, back to the rod. You see the magic of different delays. So then our brains have to understand the nature of delay and deal with them. There are problems if the delay should be wrong. Now, I'd like someone to come and help me, someone who thinks that they'd be very good at reading a poem, please. Is there anyone here who might be very good at reading? Do you think you could? Okay, come on down. What's your name?
Adrian.
Adrian. This is Adrian. He's going to read us a little poem about Christmas by Lewis Carroll. Sit down there, and can you read that? Read it nice and clear, nice and loud for everybody.
Thus, forgetting tricks and play, for a moment, lady dear, we would wish you, if we may, Merry Christmas, glad New Year.
Well, that's absolutely marvellous. That's so good, Adrian. I think since we've got the television equipment here, we really ought to record that for posterity. So if you'd just like to speak into the microphone, I'll put the headphones on so we can get you all set for the recording, all right? So, if everybody could be quiet, we'll start again. Off you go.
Thus forgetting tricks, lady dear, we would wish you, if we may...
There must be some sort of problem here, I think, Adrian. I think what we ought to do is hear what Adrian is hearing through these headphones. If we could couple the same system to me, what you can hear... Could we stop it, please? See, what Adrian was listening to was his own voice delayed by a short time, by a fraction of a second. We're always used to hearing a delay in our own voice because of the processing time is made longer than it should be, artificially, with a tape loop produced by the BBC. Then our brain literally can't function properly. Thank you very much. Sorry to have fooled you, Adrian. Well, let's turn to another piece of magic of the brain working on the senses, to the perception of shape. The most important thing that our visual system does is to recognize what objects are, to recognize their shapes. What information could we possibly use to do that? Well, the first slide shows the most obvious way in which we're able to recognize shape in patterns. Here's a familiar face, a picture of Father Christmas, consisting of black and white areas. We know what it is because of the brightness differences within the picture. Our brain interprets those brightness differences to produce a sensation of shape. Now let's look at the same thing over here and see whether we might be able to use color information to do the same thing. I've got a picture here, exactly the same picture, but it's painted in red and in green. If I turn the projectors on, turn the room lights down, please, what I'm going to do is project onto that picture through a red and a green projector. Now, if I turn the red projector down, you'll see that the pattern appears to be simply black and green, because I'm only throwing green light onto it, therefore we don't see the red areas at all. If I turn the green one down and the red one up, then we see a red face on a black background. If I set the red projector at about half strength like that, and now turn the green one up, there must be a point at which the green and the red have exactly the same brightness. The only difference between the two pictures will be color. Those of you with black and white televisions may in fact lose your picture altogether at some point as I turn the green up and down. Yet always we can see the shapes. So we're able to recognize shape on the basis of pure color differences as well as brightness differences. Thank you. We're also able to recognize shapes without brightness or color differences. Can I have the next picture, please? I think you'll be able to tell what this is straight away. It's a nice Christmas tree with decorations hanging around it. You can see its shape very, very clearly. Yet, if you look at parts of it, there are no edges at all present in them. There are some areas where there are simply no edges and no real edges at all, and yet we see them as if there were edges, merely because of the presence of these surrounding objects. Somehow our brain, in that case, is able to infer, to deduce the presence of an edge merely because of the surrounding shapes, not because of a difference of brightness or a difference of color. It can do the same thing based on the detection of movement. Could I have, please, the next piece of film, but I don't want it to start moving. I'd just like it to be a stationary picture to begin with, please. This is one frame from a movie made by Dr. Bela Yules in the Bell Telephone Laboratories in the United States. And as you can see, it's a completely random pattern of dots. There's no brightness difference, no color difference between any part of the pattern. But if we create a movement difference, now, please, instantly you can see a shape, a letter T, standing out very clearly against the background. So that shape difference is created only by the detection of a movement difference. Well, we can detect really remarkable things by analyzing shape, and I'd like you, if you would, to watch the monitors while the lights go right down to see whether you can recognize this next shape. Could we turn the room lights all the way down, please? And could you watch the television monitors, please? Now, what on earth could that shape be? Well, I think we should see what's going on, don't you, Bill? Here we are. Would you like to come in? Step down, thank you. Walk to the front. Okay, now, could we have the lights up, please? This is Melanie, who works at the Royal Institution. Thank you very much, Melanie. Melanie's wearing lights. You can see at all the major articulations, the joints of her body, just a few lights scattered around. But I think you'll agree that the information provided by the movement at those points was quite enough to identify her as a person, simply from those few dots of movement. Thank you, Melanie. Well, finally, let's ask where we get our expectations from. They're built into our brains, but are they built there genetically before we're born, or do we learn them? Well, in fact, it's a combination of the two. And the extent to which we're able to learn new expectations depends on our species. Imagine an animal that needed to have the ability to analyze its senses as soon as it was born. The next piece of film shows such an animal. A giraffe being born. Now, giraffes live in groups. They're hunted by predators, by lions. They have to be able to move and therefore to be able to use their senses very, very shortly after birth. If they didn't, they wouldn't survive. So we see here within seconds, certainly within minutes after birth, this baby giraffe being licked now by its mother will get up, will stand on its legs, will walk, and will soon start to run with the herd. So many animals, simply because of their biological needs, must have their sensory expectations working immediately after birth. But other animals, thank you very much, Bill. Other animals, like these baby mice, certainly don't have many expectations about the world. They're born very helpless, very, very immature. Their eyes are still closed. Their ears are sealed. They know almost nothing about the world. We saw that they're able to communicate with their mother by screaming in ultrasound, and in a way, their brain is simply designed to use the sense organs of their own mothers. They just communicate to her. So, shall we put them back with Mum? Thank you very much. So, the extent to which animals have their expectations built into them depends on their needs. Human babies certainly have many expectations, even at birth. The sensory system of man, as we're discovering more and more, is actually quite well developed at birth. Although, of course, there are many things that we learn through our own experiences during the first few weeks, months, of life or even later. The next piece of film shows experiments on very young babies by Klaus von Hofsten in Germany. And you'll see that this little baby is able to follow a moving object with its eyes. It must know where the object is, it must recognize it, and the baby can even reach for the object in the correct position in space as that object passes. If you watch carefully, the baby reaches out as the object goes by, there. So this baby's brain must already have built into it knowledge about its own delays of analysis, as well as an ability to interpret shapes and movements in the world. But many animals, certainly including man, nevertheless learn from their own experiences. And perhaps the best-known example of that is the phenomenon called imprinting, discovered by Konrad Lorenz. The next piece of film is going to show us the sort of experiments that Lorenz did. He discovered, you'll remember, that young geese and ducks and chickens, if they were reared by a human being or by any kind of unusual figure, would learn to follow that object, that person, as if that person were its mother. The animal would gain its expectations about the world from its own early visual experiences. Could we then have the next visitors, please, Bill? Thank you very much. So here we have some baby chicks which were born in the Royal Institution, which hatched in the Royal Institution just a couple of days ago. And their problem is that they haven't got a mother. I wonder if there's anyone in the audience who might be able to provide a mother for them. Would you like to come down? Well, coming down now is Jessica, who's my youngest daughter. And I think she might be able to provide just what's needed. Come along, Jessica. Thank you. Can I have that? Thank you very much indeed. Do you want to come forward, Jessica? Okay. Now, this is a little toy duck. And if I switch it on... Bill, there seems to be problems. Can we... toy duck. Are there any batteries there? Over the... Thank you very much. Okay. Well, when the mother gets her energy back, perhaps we can... Thank you. Let's put her down. It's still not working. Oh, never mind. There, I think even by moving it around, you'll see that the ducks, the chicks, some of them already are following the mother, even without the batteries. In fact, these chicks were reared with that model in their box. They've become imprinted on it as if it's their artificial mother. So the chicks, I can assure you, will go back to a good home when they'll learn about real chicks, not about model ones. So animals need to learn in order to see properly. In fact, all animals can learn to some extent. Virtually all animals can learn to some extent. Even simple insects are capable of learning and therefore changing their perception of the world. Now, earlier today, Dr. Peter Mobbs from Queen Mary College came in to the Royal Institution and helped me to train some honeybees. And I'd like to show you a piece of videotape, if you watch the monitors, of those bees learning very intelligently on the basis of just one practice trial. If you watch the film, here's a little bee mounted in a tube, mounted in a metal tube, and you can see its antennae waving around. What I'm going to do is blow a puff of air with the smell of carnation oil over its antennae. There we go. It's blowing now. Nothing happens, you notice. But then we feed the bee. We touch some sugar solution. You see its tongue coming out. It's proboscis. Can you see that clearly? It's licking the sugar. Now, what we hope is that it's going to learn the association between the smell and the taste. Now look what happens if, when it puts its tongue back in, I blow again on its antennae. Remember, it did nothing the first time. But now I'm going to blow the carnation odor on the antennae now, and immediately you can see the tongue coming out. The bee has learnt in just one trial the association between smell and the taste. I think we try on the videotape once more. There we go, and the bee reacted again. Well, now we've got here on the bench some bees that we trained this morning, and we'd like to look first of all at the, could you hold that for me, Max, thank you? This one over here, this one on this side, which hasn't been trained. I'm going to blow the oil now, and there's no reaction from it. But if we could move across from these three, these are the clever ones, these are the ones that know. Let's see if we can see any reaction from them. Now? Yes, there's the tongue going out. The middle one reacted. Maybe some of them have forgotten. Well, certainly the middle one's reacting very strongly. Look at that. You see the tongue coming out. He's still remembering, thank you very much, he's still remembering what he learnt even this morning. So the sensory systems of animals depend on their expectations of the world. Some of those expectations are built into the animals when they're born. Some of them are learned. And man, perhaps the supremely intelligent animal, perhaps, can continue learning very quickly about his senses all through his life. Now, I'd like some help, please, from, what's your name, please? From Tim. Tim said he would help us with this experiment earlier. Would you like to come over here, Tim? He doesn't know what's going to happen. Now, Tim, if you'd like to stand over there, I'm just going to test how good you are at throwing things. I want you to take the balls from there, one at a time, and throw them to me. Not yet. Wait till I get something to catch them in. I'm going to catch the balls in a box. Just throw them to me. Let's see how good you are. Well, that's excellent. Okay, one more. Now, do you think you could do that with a pair of glasses on, Tim? All right, let's put the glasses on now, please. Now, these are rather special glasses, as you'll see. Okay, there they are. Now, could we just try again, please, Tim? Straight in, just like you were before. Just straight into the box, please. That's not very good, Tim. Would you like to try again? Now, just carry on, please. Just keep doing it. Tim's problem is, you're getting better. Tim's problem is that he's wearing a pair of prisms. Carry on, carry on, carry on. He's wearing a pair of prisms, just keep going, which make the world seem to shift to one side. Carry on, keep doing it all the time, please, Tim. Excellent. You're getting, and again, next one. And you can see that he's correcting. He's almost completely corrected. Almost. In a few seconds, carry on, please, just carry on. Yes. Started way out here, carry on. And he's now almost exactly accurate every time. Okay, now would you like to take the, now you've almost completely corrected, take one more in your hand, ready to throw. Okay, good. Right, off you go then, throw it now. Go on. Well, you saw how he deviated to the other side this time. He was throwing there before and there afterwards. And I think he'll get perfectly accurate in a moment. Just keep going. There we are, corrected again. Thank you very much, Tim. I hope you'll agree that Tim's brain had built new expectations about the world, about the nature of the connections between vision and movement in just a few seconds. Well, I hope that you've enjoyed this trip through your own perceptual worlds and into the sensory worlds of other animals. And I'd like to take this opportunity of thanking the many people who've made this possible. First of all, of course, Bill Coates and all the staff of the Royal Institution. And then the television staff, the cameramen and the engineers who've helped me so much with so many demonstrations. Many people in my own department in Oxford, John Bannister, Lawrence Waters, John Eldridge, and perhaps most of all John Mittell, who's been here for many of the demonstrations helping with the electronics. Finally, all the people around the world who've helped me providing information for the lectures. I want to wish them and all of you a happy and a very sensible New Year.